Hello student, welcome to this channel. I am Anuradha Kashyap, your biology mentor. You all know very well that in this channel I am going to share a lot of topics which are related to your biology subject. And today in this video I am going to share a topic that is your restriction endonuclease. So first of all, before knowing about the restriction endonuclease, we have to know about biotechnology. So, students, when we go in depth with biotechnology, then it comes to know that there are a lot of bacteria, a lot of microbes which are used in industries for their own services. Like for example, if we talk about biotechnology, and when you break down these two words, that is biotechnology, like here you can see biotechnology. What is the meaning of biotechnology? So student, when we break these word in two part, one is your bio means living, and technology means technologies, or we can say techniques. When we use lot of techniques in living organism to modify them so that we can obtain our own services, our own product, then it is known as biotechnology. Basically, it is the alteration method where you are going to change a microbe so that it can give you lot of main products which are used for your own purpose. For example, I can say you biotechnology basically are of two types. One is your old biotechnology, or we can say it classical biotechnology, right? And second one is your modern biotechnology. What is the major difference between both of them? So, in topic restriction endonuclease, we have to know about these main topics of biotechnology. What is your old biotechnology and what is your modern biotechnology? In old biotechnology, we are not going to change the DNA of any microbe. We are not going to modify that. For example, there is a product that is your curd, right? In home, we already made curd. Lot of time we made curd, and we always use lukewarm milk in which some inoculum is added. We are going to add some inoculum in your milk, and after that we obtain our product that is curd. So what happening here? In your starter, or you can say in your inoculum, there are a kind of bacteria which is your lab means lactic acid bacteria, or we can say example like lactobacillus are those microorganisms which can convert your milk into your main product, that is curd. So basically, this is the process where microbes are going to change your substrate into its product, milk into its curd. Okay, done. So here we can say that in old biotechnology, basically microbes can change one substrate into another substrate, or into another product. But here, microbes are not changed. Microbes use their own natural capabilities, so that by using that, microbe can change one substrate into another product. So here there is no change in the microbe DNA composition. But when we talk about the modern biotechnology, there is a concept that is known as genetic engineering. What is basically the genetic engineering? We are we are going to engineer some uh, some new things into the DNA, into the genome of microbe. That's why it is called as a genome genetic engineering, which is a part of your biotechnology. So basically, in genetic engineering, we are going to modify the DNA sequences or genome of the microorganism so that we can obtain our own product, what we desire, right? The product which we desire, we can obtain only that product when we are going to change the genetic composition of your main genetic composition of your bacteria or your any kind of microbe. It can be your yeast, it can be your any kind of microbe. For example, it can be a fungus, it can be a bacteria, it can be a yeast. All these are microorganisms, right? So this comes under your modern biotechnology. So basically, what is the concept behind restriction endonuclease? You can you are very familiar with the term that is ase. Ase are basically the suffix which we normally use to describe any kind of enzyme. So when I talk about restriction endonuclease, so I am talking about a kind of enzyme, right? So in this video, I'm going to use a presentation which is directly taken from the Google search. It's not my own presentation. So all the references will be given in the end of the video. So in the end of the presentation, basically. So here you can see. Okay, uh, give me 1 second, students. So here you can see this is the restriction endonuclease topic, and restriction endonuclease is basically the tools inside your biotechnology in 12th class, or for the NEET preparation for students who are preparing for their NEET. There is a unit that is your biotechnology, and there is first chapter biotechnology principle and its application, and the topic restriction enzyme comes under that chapter, right? And this topic is very important for NEET aspirant, because in this topic, from this topic, at least 1 to 2 questions are always asked from this topic, right? Yes. So restriction enzyme. From here, our topic going to start. What are basically the restriction enzyme? Restriction enzyme are basically the tools inside biotechnology. So what happened during biotechnology? I told you that we are going to change genetic composition of any microbe. So for the change, for the alteration in the genetic composition of any microbes, what we have to do? We have to cut the genome of that particular bacteria or particular microbe, and then we are going to add some additional part, or you can say additional segment of DNA, which is your desired gene. So this is your bacteria, and this is the DNA of the your DNA of your bacteria, which contain their own genome, right? But you want to add something here from outside. So first of all, what you have to do, you have to make space inside the DNA of your main bacteria, or you can say main microbe, right? So in the genome of bacteria, you have to make space so that you can add DNA from the outside, right? So to cut that main particular genome, or the desired gene segment from where you are going to take it. For example, um, there is a genome, there is a gene segment for insulin, right? Insulin gene segment is present in eukaryote, but you are going to add it inside the prokaryote, for example in your E. coli. So first of all, you have to make space inside the genome of your E. coli, right? You have to make space there so that you can add your insert, or you can add your desired gene into them. So insulin is your desired gene, but eukaryotic DNA is also very big, right? Eukaryotic DNA is also very big. So you have to cut that insert, you have to cut that desired gene from eukaryotic gene. So what is the main tool? What is the main tool to cut that? So that you can cut it, with the help of which tool you are going to cut it. So that tool is your restriction enzyme, or you can say restriction endonuclease. Restriction endonuclease is the enzyme which are used to cut, or which are used to make specific cut inside the DNA segment. Inside the DNA segment, so that you can make your insert. You can release your insert from your desired gene segment, and then you can add it another site. Clear? So I am going to describe it uh, with my with the written thing, right? So here you can see what I'm going to say. For example, for example, for example, this is your gene segment, right? Hmm. Okay, look at this. For example, this is your gene segment, right? And this is your gene segment which encodes for insulin. This encodes for what? This encodes for your insulin. This encodes for insulin. Insulin gene is present in eukaryote. You know very well that it is present in eukaryotes. My pen tab is not working as such, so please excuse me for this. And insulin is your eukaryotic gene segment, right? You are going to add this insulin gene, but you in eukaryotic DNA is very big. You can consider it is a big segment of gene, big segment of lot of genes. This is your eukaryotic DNA, but you are going to you just want a segment of gene which encode for insulin. Suppose this encode for insulin, right? This segment encode for insulin. So you have to cut it from here and here, right? So that you can obtain this particular segment, that is your insulin gene. So with the help of which tool you are going to cut it? First question. Second question, there is a vector. Now this is new term, that is your vector. Vector can be your plasmid. I'm not going in detail with vector. I'm just talking it. Vector act as a vehicle. Vehicle is basically what which can transport one thing from one place to another place. This is your vector or this is your vehicle. In this case, plasmid act as a vector. Lot of another vectors can be there, but I'm talking about plasmid, right? Plasmid can act as a vector. So basically, first of all, you are going to cut your desired gene. This is your desired gene, that is insulin, and now you are going to add it into vector. Vector is basically what? It is your plasmid. Plasmid. So when you are going to add your desired gene into plasmid, or you can say vector, so first of all you have to make space in this, right? You have to make space in your plasmid. So what happened? You have to cut it. You have to cut it. You have to cut your plasmid. So again, with which tool you are going to cut it? So this is your second question. So both kind of DNA, whether it is your desired gene, whether you are going to cut your desired gene from a eukaryotic DNA segment, eukaryotic DNA, and you are going to cut plasmid or vector to make space so that it can ligate with your insulin or with your desired gene segment. So both type of DNA, plasmid is also a double stranded circular DNA. So both type of DNA are cut with the same tool in your biotechnology, and that tool is your restriction endonuclease. This is a kind of enzyme which can make a specific cut in your main DNA or in your plasmid, right? Okay. So basically, from where we can obtain restriction endonuclease. Now we will deal with the different questions. First question is here, from where we can obtain restriction endonuclease, right? Second question is why restriction endonuclease are produced in that species. Second question is your that. Third question, how many restriction endonucleases are obtained till now? How many restriction endonucleases are obtained from different strains of your bacteria till now, right? And fourth, which kind of ants they produce? It can also your fourth question. With what will be the nomenclature of your restriction endonuclease? So all these questions will be answered one by one in this video. So stay tuned with me. Now I'm going to sit at presentation, right? So what is basically the restriction enzyme? Restriction enzyme are a kind of enzyme which can make specific cut in your main DNA. You just have to listen me carefully. Restriction enzyme are those enzymes which can make the specific cut in between your DNA segment. For example, here you can see, okay, restriction endonuclease are produced inside the bacteria mainly. They always produced in prokaryote. Prokaryote is basically what? They are basically bacteria. So restriction endonucleases are mainly produced inside the bacteria, and they never produce inside a eukaryotic cell. We are eukaryote. We don't have any kind of restriction endonuclease, right? So first of all, why they produce inside a prokaryote? There is a defense mechanism. Mark my word, there is a defense mechanism inside the prokaryote due to which restriction endonucleases are produced inside a prokaryote. For example, there is a here you can see. For example, there is a E. coli. E. coli is what? E. coli is a prokaryote, right? There is a E. coli. And this is the main genome of E. coli. Consider it as a main genome of E. coli. Consider it as a main genome of E. coli, right? Consider it as a main genome of E. coli. Done. Now, there is a kind of virus which is called as bacteriophage. What is bacteriophage? Bacteriophage is a kind of virus which can feed upon bacteria. Okay, bacteriophage comes and stick to the cell wall of the bacteria. This is your bacteriophage, which can feed upon the bacteria. It is called as bacteriophage, and it insert their DNA into the, or we can say it insert their genetic material inside the prokaryote, inside the E. coli. This is the genetic material of this bacteriophage, and now it is going to add their genetic material inside the prokaryote. And this genetic material of bacteriophage are now insert itself inside the main genome of prokaryote, due to which the all over the machinery of prokaryote will be hacked by the bacteriophage, and now your E. coli will going to add, because no proteins will made for E. coli. All the proteins will be formed for bacteriophage. That's why E. coli cannot survive. E. coli will not able to survive, and death of E. coli will happen. Right. So this is the thing which always happens with a lot of bacteria, right? Lot of prokaryote. So in response to that, prokaryote started to defend themselves with the help of a mechanism which is basically production of two kind of enzyme. Again repeating, there is a defense mechanism inside the E. coli to defend itself from the bacteriophage attack by producing two kind of enzyme. So what are these enzymes? Okay, again, here you can see this is, sorry, this is what, this is again E. coli. Okay, so for example, this is again E. coli, and this is the main genome of E. coli. Now what happens? Bacteriophage attack again. Bacteriophage attack happen again. Here, bacteriophage attack again and insert their main genome inside the what? Inside the E. coli. Okay. Now, in response to protect themselves, what will happen? This prokaryote already formed two kind of enzyme in their own body, and these two enzymes are, one enzyme is, for example, this is one enzyme, and for example, this is second enzyme, right? So this one enzyme is methylase, and the second enzyme will be restriction endonuclease. So, ma'am, what is the function of methylase? Now, this question comes in your mind. What is the function of your methylase? Okay, this is pretty much clear that what is the function of restriction endonuclease. It can cut the it can cut the DNA so that the desired gene can be obtained, right? So, what is the function of methylase? And the second question will arise in your mind: if restriction endonuclease is produced by this bacteria, then this restriction endonuclease can cut their genome itself, right? This restriction endonuclease can work on this genome also and chop up it lot of segments. So lot of segments can be obtained by this restriction endonuclease inside the prokaryote. So what is the criteria behind the formation of this restriction endonuclease inside a prokaryote? What is the main function basically here, and what is the process going on? How it works? And what is the main motive to produce methylase enzyme inside a prokaryote? So here you can see here two enzyme methylase, restriction endonuclease. As the bacteriophage DNA come inside the prokaryote, this restriction endonuclease going over this and this bacteriophage genome and scan the DNA. What it will do? Restriction endonuclease will scan the DNA of bacteriophage and find out a palindromic sequence. Now, what is a palindromic sequence? Is I am going to tell you in the by showing you by showing the presentation. And restriction endonuclease, what it will do? Restriction endonuclease first of all scan the complete DNA of bacteriophage and then find a site which is the palindromic sequences, and this sequence is known as recognition sequence. Within the recognition sequence is basically 6 to 8 base pair, right? 6 to 8 base pair. This is the recognition sequence. And now restriction endonuclease find out this recognition sequence and make a cut at the specific site, that is the restriction site within the recognition sequence. I hope this is clear. All over the DNA, it scans all over the DNA. And when it scans all over the DNA, there is a sequence which is the palindromic sequence, which is called as recognition sequence. And within the recognition sequence will be 6 to 8 nucleotide base, 6 to 8 base pair long. And within this recognition sequence, there is a site which is restriction endonuclease site, and the which is the restriction site, sorry, which is the restriction site, and restriction endonuclease enzyme recognize that restriction site and make a specific cut at that position. This is a function of restriction endonuclease. Now, the second question is, if restriction endonuclease can make specific cut at the DNA, then why it cannot cut the main DNA of main DNA of bacteria? This is the second question, right? And what is the function of methylase? Basically, restriction endonuclease enzyme can work on only on that DNA which is unmethylated. Okay, again I am repeating, restriction endonuclease can make specific cut only at that position, only at that DNA, only on that DNA which is unmethylated. If DNA is methylated, so restriction endonuclease cannot recognize it and cannot make a specific cut there. Is that clear? Okay, so now I think that it is clear to you what is the function of methylase here. Methylase is the second enzyme which are going to add methyl group on the main genome of the bacterial DNA. First of all, what it will do, methylase, the enzyme which is produced by bacteria itself, this methylase enzyme are going to add methyl group on this DNA. And now restriction endonuclease cannot recognize this DNA. Now restriction endonuclease can only recognize the outsider DNA, foreigner DNA, and make a cut at the specific site inside the bacteriophage DNA, and due to which bacteriophage cannot make their own protein and it will die. It will die. So bacteria can protect itself with the help of this defense mechanism. That's why this this restriction endonuclease are obtained only from prokaryote, not from your what? Not from eukaryote. Okay. So first of all, there are three scientists who discovered the restriction endonuclease from the E. coli in 1963. I'm going to repeat, in 1963. There are three scientists, Arbor, Smith, and Nathan, who discovered the restriction endonuclease from the E. coli. E. coli was the first bacteria from where this enzyme was discovered, and till now 900 restriction endonucleases are discovered from 230 strain of bacteria. This is the main question of NEET. Lot of times this question can be asked. This question was asked that how many restriction endonucleases are discovered till now. So according to your NCRT, till now 900 restriction endonucleases are discovered from the 230 strain of bacteria. This is clear to you? Okay. Now, when here you can see Okay, there is one another concept which is very important. According to your NCRT, when we talk about restriction endonuclease, so from which class this enzyme will belong? So in your biomolecule chapter, there are six classes. Six classes are mentioned of enzyme. So there is one class that is your nucleases. Nucleases. Nucleases is the biggest class, or we can say nuclease is a big class of enzyme, and this is the class which act over the nucleus, or we can say which act over the genetic material. Either it can be RNA or either it can be DNA, right? Nuclease class is further divided into two subclasses. One is your exonuclease and one is your one is your endonuclease. Student, nucleases have two subclasses. Nuclease is a big class. It has two subclasses. One is your exonucleases and one is your endonucleases. So if enzyme work over the edges, for example, at the outer side of the DNA, either it can be left or either it can be right. For example, either the enzyme is going to work on right corner, or it work over the left left corner, then enzyme work over the edges of the DNA, not in the middle of the DNA. Okay, so this is the exonuclease. When any kind of enzyme work over the edges of the genetic material, then they are considered as exonuclease. But when these enzymes are act inside the middle of the genetic material, middle of the DNA or either or RNA, so they are considered as endonucleases. They are considered as what? They are considered as endonucleases within the DNA, or we can say in middle of the DNA. When any kind of enzyme are going to work, then it is called as endonucleases. And when I'm talking about restriction endonucleases, it means I'm talking about the enzyme which work inside the DNA or in the middle of the DNA. It can make specific cut only inside the DNA, inside or in middle in the DNA. Okay, now there are lot of types of restriction endonuclease, which are basically some advanced material, not in your NCRT, not for NEET student. I'm just going to explain it uh, in brief. Okay, so basically there are three kind of restriction endonuclease: type 1, type 2, type 3, right? Type 1 and type 3 are the enzyme which require, they are the types of restriction endonucleases only. They are the type of the restriction endonucleases. They are not the type of endonucleases. They are not the type of exonucleases. They are the type of the restriction endonucleases. There are three types. One is type 1, type 2, and type 3. Type 1 and type 3, they are not in the genetic engineering. Why? Because they require magnesium ion for their function. They also require ATP for their energy and additional cofactor, that is where as adenosyl methionine, which is also written as the same in brief, in abbreviated form, right? So as adenosyl methionine, ATP and the magnesium ion are the requirements which are used by the type 1 and type 3 kind of restriction enzyme. But type 2 are the enzyme which required only magnesium ion. Magnesium ion, it doesn't require ATP or SAM. So it is easy to handle. That's why type 2 is very commonly used inside the genetic engineering, inside the biotechnology as a tool. Type 2, it is most common. Type 1 and type 3 are the enzyme which are not going to cut at the specific position. For example, there is a recognition sequence, so we can say it is a palindromic sequence. I'm going to tell you that what is a palindromic sequences. For example, there is a DNA, and in the DNA there is a palindromic sequences which is also called as recognition sequence. We have to make cut at that restriction endonucleases, but we have to make cut at that restriction site. Okay, but when we use type 1 enzyme, type 1 restriction endonucleases, so they are not going to make cut at that restriction site. They will make a cut at 1000 position away from the main DNA, away from the desired gene, away from the recognition sequence. They are going to make a cut. They are going to make cut at the 1000 base pair away from the recognition sequence. So you can think about it. This is not a good idea. This is not a good thing. It is just a wastage of thing. So type 1 is um, not at a prior position inside the endonucleases. Type 3 is also do same thing, is also kind of do a kind of same thing. For example, this is a recognition sequences inside the DNA, and we have to make cut at the restriction site. But this type 2, type 3, sorry, this type 3 restriction endonuclease scan all over the DNA and find a recognition sequence at 100 to 500 base pair away from the recognition sequence, which is the main recognition sequence. So again, this is just a wastage of energy, wastage of iron, wastage of time. So type 3 and type 1 are not considered as a suitable enzyme inside the genetic engineering. But the type 2 is the main enzyme which is most specifically used because it make a specific cut at the palindromic sequences, or we can say it make a specific cut at the particular restriction site where we have to where we want it, where we want to make a specific cut. Okay, so that's why type 2 is most suitable, we can say tool, most suitable tool in your biotechnology. So here I am going, I already written type 1 and type 3, type 1 and type 3. This is type 3, not used in genetic engineering. Okay, type 2 is the specific, and type 2 is the specific and most commonly used. Okay, now switch to presentation. This is written here. What is restriction enzyme? They are the protein. Basically, what are enzyme? Enzyme are the proteins. You all know very well that the DNA is going to encode itself inside the mRNA, which is your transcription, and from the mRNA we obtain protein, which is by the method of translation, and protein gets converted into a lot of different enzyme when it folded over and make tertiary or quaternary structure, make active sites inside it, then it leads to the formation of your enzyme. So basically restriction enzyme, these are the protein produced in a bacterial cell as a defense mechanism which can make cut a specific site. Okay. They are also known as restriction endonuclease or restriction enzyme. We can use these to manipulate DNA in the lab. We are going to work with these restriction enzyme inside the laboratory level. Okay. Now I will tell you about the palindromic sequences and nomenclature within a few minutes. First of all, I am going to tell you why don't bacteria destroy their own DNA with their restriction enzyme. I already told you that in the bacteria there is a restriction, there is a defense mechanism. For example, this is a bacterial host cell, and this is the host DNA which is mentioned here, represented here in blue color. And this is the bacteriophage, the bacteria, the viruses which can feed over the bacteria. It stick to the, it adhere to the bacterial cell wall and insert their own DNA inside the bacterial cell. Now, in response to that, for their defense mechanism, host DNA produces two kind of bacteria. One is your methylated DNA, one is your methylase, and the second one is your restriction enzyme. What is the function of restriction enzyme? Restriction enzyme will stick over the outsider DNA or bacteriophage DNA. In green color, restriction enzyme is representing. This restriction enzyme will stick over the bacteriophage DNA and chop it, right, into several fragments. So basically, the phage DNA will degrade it. Now, question comes in mind that why this blue color main genome DNA is not cut by restriction endonuclease. So here you can see these are the group, these are the methyl group which basically block the restriction endonuclease, which block the function of the restriction endonuclease, because this DNA is not recognized by the restriction endonuclease because methyl group is added here, and this protect the bacterial DNA from being cleaved. That's why bacteria destroy their own DNA. That's why bacteria never destroy their own DNA with their restriction enzyme. I hope this is clear to you. Okay. Now, What is basically happen here? What is the main function of methylation? Here you can see when there is no methyl group in over the DNA, this is your unmethylated DNA. Methylation is not here, methyl group is not added here. So basically restriction enzyme come and recognize these palindromic sequences and then make a specific cut inside them, right? And then make these thread ends, which are known as the sticky ends. I will tell you what is the sticky, what are the sticky ends, what are the blunt ends. Just wait, okay? Now this is the unmethyl again this is unmethylated DNA. No methyl group is added here, but there is an enzyme which is methylase. Now this enzyme will add methyl group here. Here you can see over the base that is adenine, methyl group is added. Okay, here methyl group is not added, but here methyl group is added. Now this is a methyl group. Now eco R1. Now restriction enzyme comes here and it will not recognize this methylated DNA. This is the methylated DNA and it cannot recognize the methylated DNA, and eco RI will not claim methylated DNA. So this is the main protective function of the methylation inside the bacterial DNA. Okay. Now what are the nomenclature, how we can give name to the restriction endonuclease? So for that, here you can see. Okay, here you can see, for example, when I'm talking about the nomenclature of what nomenclature of restriction endonuclease. Suppose, as I told you that all restriction endonuclease is obtained from the prokaryote. Suppose I have a restriction endonuclease which name as eco R1. Okay, so what it represented, what it is going to represent. First letter, as it is in the capital alphabet, it is a capital E capital alphabet. So it is going to represent a genus. According to Carolus Linnaeus, there is a binomial nomenclature. So what it is genus. Second two words are written in small letter and they are going to represent a species. So here genus is Escherichia. Escherichia. CO representing coli. So it means it is obtained from Escherichia coli, and R is basically representing strain, strain or dye, any kind of dye, and I, it is not I actually, it is number, which is first number, Roman letter, in Roman letter first number, which representing your order number or discovery number or um, the enzyme number which which are used in lab. Okay. So this is the nomenclature. First word representing genus, second two word representing species, one word is representing strain, and one number is representing this order number. So this is the nomenclature of RE. For example, there is another example that is Hind 2. Hind 2 was the first restriction endonuclease which is discovered from the E. coli, and this was discovered by basically, it was discovered in 1972, or we can say 1972 by Stanley Cohen, and discovered by, sorry. Hind2 is the enzyme restriction endonuclease which is discovered for the first time in the bacteria from the prokaryote, but its function is characterized later after 5 years later. Its function is characterized after 5 years later. Okay, so Hind 2, what is it? What it is representing? Hind, first word is representing genus, that is Haemophilus, and second two word is going to represent species, that is Haemophilus influenza. Haemophilus influenza. D is representing again a dye, and 2 is representing the order number. Okay, if I'm talking about, for example, this SMA1. What is the meaning of SMA1? S means, as here, S is representing Serratia, MA representing here Marcy sense, okay? And 1 is representing here order number. You can check the spelling also. And the fourth one, for example, I can take another example, balm H1. So here balm B is representing genus, that is Bacillus road-like bacteria. AM is going to represent amyloliquefacial. Amyloliquefacial. And H is going to represent your strain, and the fourth one first is representing your order number. So this is about the nomenclature of restriction endonuclease. And I will tell you about the ends, sticky ends or blunt end. What are these? So here you can see, and one question comes in your mind, one question can come in your mind, that I'm talking about that restriction endonucleases are going to make cut at the specific position, but basically at which side they are going to cut, which side they are going to cut in the DNA. So here you can see they are not going to work over the hydrogen bond, they are not going to work over the glycosidic linkages, they are going to work only over the sugar phosphate backbone of the DNA, they are going to make specific cut. So this is a question which is previously asked into the NEET, that what is the main site, what is the main bond which is broken by restriction endonuclease. So always remember, restriction endonucleases only work over the sugar phosphate backbone of the DNA. It neither break your hydrogen bond and not break your glycosidic bond. Here, so this is the thing. So here you can see. Restriction endonucleases are also known as molecular scissor. Arbor, Smith and Norton. First of all, from here we are going to start again. Endonucleases are used which cut at palindromic sequences. Now, what is palindromic sequences? Palindromic sequences are the sequences which are same from the both hand. When we read them from different hands, then from both hand they are same. For example, Malayalam. When you start it from left to right, or you are going to start it from right to left, the spelling will be same. Malayalam. Okay, madam, or lot of words are there. So, like for example, Kanak. When you are going to read it from one hand to another hand, and you are going to read it from another hand to one hand, then both hand will be read in the same fashion. Okay, so these are known as palindromic sequences, and the recognition sequences inside a DNA over which is scanned by restriction endonuclease. That recognition sequences is palindromic sequences, or the sequences which can read same on the both sides. Okay. Now, these palindromic sequences are used by this restriction endonucleases. 930 restriction endonucleases from 230 strain of bacteria are discovered till now, and this was first time isolated by Arbor, Smith and Nathan, and they were awarded by Nobel Prize in 1978. Nathan also gave them nomenclature in 1963. First of all, they are discovered from E. coli as a defense mechanism, as I already told you. Two enzymes are produced. One is restriction endonuclease. Second one is your methylase. Methylase add methyl group to the bacterial DNA, so that restriction endonuclease cannot recognize them. I am little much fast. Okay, because I already told you everything. I'm just repeating it. I'm just reading it. Okay, so I'm little much faster. Restriction endonuclease, they are the enzyme which can recognize palindromic sequences, which is also known as recognition sequences, and make a cut there. Okay. Now there are three kind of restriction endonucleases: type 1, type 2, type 3. Type 1 and type 3 are not commonly used in the genetic engineering because they are using a lot of energy, lot of cofactors, magnesium ion. But type 2 are the most common restriction endonuclease which is used in the genetic engineering. First time discovered restriction endonuclease was Hind 2, and about when we talk about the nomenclature, first word representing genus, second two word representing species, third word is representing die, and the fourth one will be your isolated number, or you can say order number, strain number, lab number. Okay, so Haemophilus influenza RD2. And now, how restriction endonucleases work? They break phosphodiester bond of both strand. They work over the phosphodiester bond of both strand and going to make a specific cut there. Right now, here you can see. Okay, this is the DNA. Now you have to remember the particular sequences, particular recognition sequences, palindromic sequences of some restriction endonuclease. For example, in case of eco R1, you have to remember about the palindromic sequences, which is your GAATTC, which is a six base pair recognition sequences. GAATTC. This is your recognition sequence, and there is a restriction site in between the recognition sequence, and that is your between G and A. For example, here you can see this is your DNA segment, which is a palindromic sequence, GAATTC. Here you can see GAATTC. So this is your palindromic sequences. Now sorry, again I'm repeating it. Eco RI. This is Eco R1. Eco R1 recognize this palindromic sequences and now make a cut at restriction site between G and A. Here you can see this arrow indicates the restriction site where Eco R1 cut sugar phosphate backbone. So this is the restriction site on both strand. Here this is G and A, here in between G and A. So you are going to make two specific cut. And these cut are the sticky cuts. They are not the blunt end, they are the sticky cut because they can fit into each other easily. So these are known as staggered or cohesive or sticky or overhang structure. We are going to call it staggered and cohesive and sticky and overhang structure. But when we talk about the another restriction endonuclease, every restriction endonuclease can recognize their own specific restriction site. Remember it. Every restriction endonuclease can recognize their own specific restriction site on specific recognition sequence. Okay. So SMA1 have a recognition sequence that is GGGCCC. GGGCCC. This is the palindromic sequences for Serratia marcescens, and there is a restriction site between G and C for Serratia marcescens. But this restriction site after making cut, we will obtain flush ends, blunt end, non-sticky end, non-cohesive ends. These are the ends which cannot easily fit into each other. They cannot fit together. That's why they are not much preferred. Much preferred restriction endonucleases are those enzymes which can give you specific cut, which can give you staggered cut, which can give you sticky ends. Okay, so eco R1, these are the enzyme which can make a staggered cut. But SMA1 is not as much preferred. Here you can see. Okay. Here you can see hint 2. Hint 2 have a restriction site, recognition site, that is GTT-AAC or GTT-AAC. So this is the recognition sequence for hint 2, right? Basically recognition sequence is always 5 to 6 base pair. So this was all about the restriction endonuclease. Now I'm going to represent it with the help of presentation. Here you can see I told you that what is the restriction endonucleases. Now I told you about the naming and discovery that they are produced from the bacteria as a defense mechanism. What will be the process of their nomenclature and how much restriction endonucleases are discovered till now. So this is a nomenclature process. Here E representing Escherichia, CO representing coli, R representing strain, and order discovered is 1. So this is the basically desired gene which is fragmented with the help of enzyme restriction endonuclease is eco R1, and this dark black color representing vector DNA. Vector DNA are always cut with the same restriction endonuclease enzyme by which desired gene is already cut. Okay, so for cutting off both kind of DNA, we are going to use this restriction endonucleases. Now sticky ends are produced and they can easily attach with each other with the help of DNA ligase enzyme. This is the restriction enzyme action of E. coli. Now this is the recombinant DNA. This is the recombinant DNA. Okay. I told you that the restriction endonucleases firstly scan the DNA and find out a palindromic sequences. What is a palindromic sequences? For example, Abel Vazai, Ire, Iso, Alba. If you are going to read it from left to right or you are going to read it right to left, then it will read as same. Okay. BAM H1 site GGATCC. GGATCC. If you are going to read it from from 5 prime on both strand, right? Now restriction endonucleases produces two kind of ends, sticky ends and blunt end. Sticky ends, for example, produced from eco R1. GAATTC is the palindromic sequences for the eco R1 and GA is the restriction site for the eco R1. And when we talk, so this produces sticky ends or cohesive ends. But when we talk about the site of PBU2, there is a site of palindromic site that is CAGCTG, and there is a restriction site between G and C which produces always blunt end, which is not preferred in biotechnology. Okay. Now, what are the blunt end? Here you can see this is a float. This is a chart about the different kind of restriction endonucleases, which are going to produce a different kind of ends. Cleavage patterns are showing basically here. Eco R1 will give you sticky ends. Hind 3 will give also sticky ends. BAM H1 will also produce sticky ends. Tau 3 also produces sticky ends. Hey 3 will not produce a sticky and it will produce blunt ends. So here you can see all of the recognition sequences are not always remembered, not always learned. They are just have to keep in your mind. Okay, you are not going to stick it. You just have learn. You have to learn only one sequence, that is for eco R1. That is your what? That is your GAATTC. This is a palindromic sequences which is asked many a times inside in a NEET. This is a GAATTC is a palindromic sequences, and restriction site is G and between G and A. Gattak, Gattak. This is the recognition sequences GAATTC Gattak. This site is um, for palindromic sequences for eco R1. I hope this is clear. Okay. Now these are the sticky ends. Okay, this is the desired segment and this is the cut ends of vector, which are cut by the same restriction endonucleases as it is cut by the desired gene, as it is cut as the desired gene is cut by the DNA, by the restriction endonuclease. Okay, so when we combine all these things with the help of DNA ligase, they are going to make a recombinant DNA. What is the function of methylase? I already told you. Okay. What are the example of uses? Now enzymes are used in the DNA fingerprinting. When we are going to fingerprinting in a crime scene, we are basically using DNA fingerprinting for parenting. For parenting of a child, DNA fingerprinting is used. In molecular basis of inheritance chapter, DNA fingerprinting is well explained. Okay. DNA typing, when you are going to make a profile of DNA, that then it is used. DNA sequences, when you are going to sequence a kind of DNA that which base pair comes first and which base pair comes later. This is your DNA sequencing. When you are going to splice a gene, when you are going to cut a gene, that is your gene splicing, and you are going to make a recombinant DNA. And when you are adding a vector with the desired gene, that is your recombinant DNA inside a host, that is your transformation. I'm not talking about in detail all about this. Human Genome Project is a project inside which human genome is completely sequenced with the help of this with the help of the use with the help of using these enzyme restriction enzyme. Okay. So here you can see for transformation, first of all, you are taking a desired gene, for example, human DNA cleaved with the eco R1. For example, we want a desired gene insulin. Okay, so this is the insulin gene, and suppose we want to obtain it in a large amount from plant. Okay, so we are adding into a corn DNA which is also cleaved by the same restriction endonuclease, that is eco R1, and then due to presence of complementary base pairing, they will complement with each other, and with the help of enzyme DNA ligase and using ATP, we are going to make a recombinant DNA molecule. And after this, we are going to add into we are going to add it into host DNA. Host DNA will produce a lot of lot of copies of this product. And now after removing this product, we can amplify this with the help of a technique, that is gel electrophoresis. And PCR, we can use PCR for the amplification. And for obtain desired gene segment, we can use gel electrophoresis. All those techniques will be discussed in the later section or in the later in the upcoming videos. But till now, in this video, I'm going to talk about the restriction endonuclease. So this was all about the restriction endonuclease. So after this, you can use restriction endonuclease for RFLP, restricted fragment length polymorphism, which is a different kind of thing, which you will read in higher classes. Restriction endonucleases also make a specific cut and give you lot of different kind of length of DNA, digested DNA, undigested DNA. I will tell you everything about in gel electrophoresis technique. Okay. Um, VNTR, that is variable number of tandem repeats you are going to obtain with the help of restriction endonuclease. This is the main gel electrophoresis DNA cut with restriction enzyme. So here you can see the orange band of DNA, undigested DNA or digested DNA. Leave it. I will explain it. I will explain everything in gel electrophoresis presentation. So for today, this was all. Sorry. So for today, this is all about your restriction endonucleases. I hope this is clear to you. You know very well now that what is restriction endonucleases, how they work, why they obtain, how they work, and from where they are obtained. What is the defense mechanism occurring in prokaryote? Why they are work on our DNA, because we are eukaryote and our DNA is not methylated, so they can easily cut our eukaryotic DNA, or they can cut on prokaryotic DNA also, where when that DNA is unmethylated, but due to methylation that DNA is protected inside the bacteria. Okay, so thank you students, I hope this is clear to you. And if you want to join me, so you can join me on Telegram also. If you want to PDF, if you want to PDF of my notes, because you know very well that lot of channel is also lot of like you know very well that I have one channel also, like for example easy neat crack. This is my another channel. You can follow me there also. And if you want PDF of my notes, then you can follow my Telegram channel. Okay, so first so I just want to say that you can blindly follow me on YouTube on my both channel, on this channel also, and you can follow my easy need crack channel also, where I'm going to upload videos for NEET students, basically the NEET students. This channel, my this channel is only for like a complete audience, a whole audience, like you can say a big audience. For a big audience, videos are going to upload on this channel. But if you want to specifically study for NEET, so you can refer that channel, that is easy NEET crack. You can refer that channel. And students, I hope you are liking the way, and you want to, if you want to like motivate me, then you can comment me. If you want, then you can comment me the for the next video, so that I can make lot of videos on that topic. You can give me suggestion, you can give me feedback, you can like me, and you can subscribe my channel if you want to lot of videos in future. Okay, so today we are going to wrap it. Okay? Thank you.